Our next speaker will be Dr. Lev Becker from University of Chicago, who will be speaking on neutrophil elastase and anti-tumor effects. Dr. Becker's laboratory combines proteomics, bioinformatics, and functional methods to investigate the roles of innate immune cells in cancer, obesity, diabetes, and atherosclerosis. His laboratory leverages this multi-disease and disciplinary approach to elucidate novel mechanisms and to develop therapeutics that are both efficacy and safe. He has won a number of awards and publishes in very high journals. I would read some of these here, but for the sake of time and to give him the most amount of time to present his outstanding research, I will just welcome him to the stage. Thank you, Dr. Becker.
Thank you so much for that kind introduction. And I'd also like to thank the conference organizers for inviting me to speak here today. This is my first time at this conference, and I've really been enjoying it. So the story I'm going to tell you about today is about neutrophil elastase. It's a neutrophil-derived serine protease, which can selectively kill cancer cells and attenuate tumorigenesis. Almost all of the data I'll be sharing is published back in 2021, so I'll invite you to read that paper if it's of interest. Before I begin, I have several disclosures to report. All right, so when you think about developing a broad cancer therapy, we really have to think about two daunting challenges. Number one, because cancer cells are similar to non-cancer cells, treatments need to be challenged to be selective. And second, because of the extraordinary heterogeneity observed in cancer, we need to develop treatments and therapies that are agnostic to the specific genetic abnormalities each cancer cell type possesses. And when you take a step back and think about those two daunting challenges, our innate immune system really evolved to overcome them. Being able to eliminate viruses and bacteria and parasites, we're talking about orders of magnitude more heterogeneity than what's observed in cancer. And for the most part, our innate immune system is able to eliminate these diverse pathogens while limiting host toxicity. Now, one of the key effectors of innate immunity are human polymorphonuclear neutrophils, and they're known to kill a wide array of genetically diverse pathogens, and there's evidence to suggest that they can also kill cancer cells, such as these two citations shown below. However, the mechanisms by which they're able to kill cancer cells are incompletely understood. So what we decided to do was to start isolating neutrophils from the blood of healthy donors. And there we go, it's working. And you can see by flow cytometry, we have a very clean population, and we can also validate that they're neutrophils by H&E staining, their sort of characteristic multilobular nucleus. And then neutrophils have a very short half-life, so as soon as you isolate them, they rapidly start cycling to apoptosis. And you can see here at 6 hours, you start seeing an XNA5 positivity, and you start seeing nuclear condensation. And so what we refer to as PMN media are neutrophils that have been freshly isolated, allowed to die, and during that death have released all of the contents, proteases, small molecules, into the media, and that's our starting material. So we first started by putting this media onto TNBC cancer cells and as a control, a non-cancer cell type, in this case, bone marrow-derived macrophages. And this is a 2-hour time-lapse video. I'm not sure how easy you can see, but the cells are starting to bleb, they're detaching, and within 2 to 4 hours, the entire plate of cancer cells can get eliminated with a dose of this neutrophil media. In contrast, when you put that same dose onto these macrophages, these are motile cells, but there's just no evidence of toxicity to them. They're behaving as if nothing has been added to their media. So beyond those two specific examples, we went on to look at 35 different types of cancer cells spanning 11 anatomical locations. And these are genetically diverse. There's a peppering of mouse and human cancer cells in here. And what I'm hoping you can appreciate is that this media derived from neutrophils is able to effectively kill all of these cell types. And if you look over on the right, these are normal cells, which are, for the most part, primary cells isolated from humans and mice. And we just don't see that same level of toxicity at all to them. So there's a very wide window between the ability to kill a cancer cell versus toxicity to a normal cell. That's, of course, in vitro, and there's lots of agents that can eliminate cancer cells in vitro. And so we wanted to see whether the same was true in vivo. And so working with Dr. Jeff Green at the University of Chicago, he had maintained a really nice TNBC PDX model. We injected this neutrophil media into the tumors and could show attenuation of tumor growth as well as in the induction of markers of death. And as a control, we injected the same material into a non-tumor bearing mammary fat pad, and we could not observe markers of cell death, nor did we observe any change in mammary fat mass. Now, one thing I'd like to point out is that these are human neutrophils, and when we tried this with murine neutrophils, we were unable to observe this killing, not in vitro and not in vivo. In the paper, we went on to delineate the mechanism, but I don't have time to share that with you today. Okay, so clearly we wanted to understand what within this media is mediating this selective cancer cell killing. And so in preparing it for fractionation by FPLC, we noted that when we put it through this 0.2 micron filter, you could see you could completely wipe out the ability of this media to kill cancer cell without changing protein levels whatsoever. And this, of course, suggests a selective depletion of the bioactive proteins. So we ran mass spectrometry on the sample pre and post filter, and not surprisingly, only 2 proteins were significantly depleted. One was eosinophil cationic protein. This is a pore-forming protein. And the other is neutrophil elastase, or ELANE, which of course is a serine protease. One thing I'd like to remind you of is that T cells kill cancer cells through the concerted action of a pore-forming protein by the name of perforin and a serine protease by the name of granzyme. So again, in the interest of time, I'm just going to summarize the data. We've delineated that neutrophil elastase, or ELANE, is the primary anti-cancer factor. We could show it's both necessary and sufficient. And we could also show that the amount of neutrophil elastase activity that a normal, healthy donor has correlates with its ability to kill cancer cells. Okay? And so next we wanted to understand how is elastase killing these cancer cells. Now, I'm not trying to make you squint at the screen here. You can see there's a large number of assays looking at survival, stress, and apoptosis. It's more just to give you context of where this heat map came from. My graduate student hates this heat map because it ended up being figure 4A in the paper and took her all of this work on the left hand of the screen to actually complete. But what you're looking at here are TNBC melanoma or lung cells, and there's human and murine versions. And what you can see is that elastase suppresses survival pathways. You see reduction in phospho-ERK, JUNK, phospho-NF-kappa B, see induction of stress pathways, gamma H2AX, indicative of DNA damage, mitochondrial ROS, and as well as activators of apoptosis. The other point I'd like to make is that even though these cells are quite genetically diverse, you see very similar pattern of signaling within them, and importantly, that pattern is very different from the non-cancer cells, which of course are not dying from this treatment. Now, this constellation of signaling is not common. Usually you see 1 or 2 events take place, but there's a whole lot of signaling going on. However, it was previously reported by Marcus Peters' group, and what he was doing was using shRNA to lower CD95 or the FAST receptor on cancer cells. And you can kind of see here the survival pathways, mitochondrial ROS, DNA damage, those effector caspases. So he was observing a very similar pattern to what we were, leading us to think that perhaps neutrophil elastase is working via CD95. Now, I'm sure many of you are familiar with the FAST receptor, which gets activated by the FAST ligand. Elastase doesn't work on even the extracellular components. It actually proteolytically liberates the death domain on the cytosolic side of this molecule. And what we could show was that if we decrease CD95 levels, we could protect cancer cells from ELANE. And if we increase them, we could accelerate killing. And even more excitingly, if we force expression of the proteolytic fragment of this death domain liberated by elastase, we call this DD ELANE, that was sufficient to selectively kill cancer cells independent of the presence of the enzyme. And so here's another one of those heat maps, only this time what you're looking at here are cells that were forced to express this death domain liberated by neutrophil elastase. Okay, so the fact that this death domain expressed in cancer cells killed them, but still didn't kill non-cancer cells, prompted us to think that maybe there was something downstream of CD95 death domain release that might be at play here. And so we decided to use the death domain as bait in an IP mass spectrometry experiment to ask, maybe this is binding to some factors that are high in cancer cells, but low or absent in non-cancer cells. And to our surprise, we saw this whole host of histone H1 isoforms, which were binding to the death domain in cancer cells and much less so in non-cancer cells. And indeed, roughly 1 hour after cancer cells are, in this case TNBC cells, are treated with neutrophil elastase, you see that histone H1, which is normally in the nucleus, rapidly exits the nucleus. And that is just not observed in the non-cancer cell line MCF10A. And proximity ligation assays showed that indeed this histone H1 was forming a complex with CD95, which ultimately ended up in the mitochondria. You might not be able to see it all that well, but that yellow image is a co-localization of the complexes with the red mitochondria. Our work is not the only work implicating histone H1. There was a previous paper published way back in 2003 showing that ionizing radiation caused H1.2 to leave the nucleus and travel to the mitochondria to induce apoptosis through mechanisms that were incompletely defined. And what we found was that histone H1, if we lower it in the cancer cells, we can protect them. And if we raise it in non-cancer cells, we can start making them susceptible to killing. And so, this enzyme can kill many types of cancer cells in vitro, so we also wanted to test that in vivo. We delivered it intratumorally because it's a serine protease and blood is loaded with serine protease inhibitors. But you can see across many different types of models, it attenuates tumor growth. And we also observed in all of the immunocompetent models that tumor CD8 effector T cells were elevated, and we could observe CD8 T cell mediated abscopal effects across all models tested. Now, we were a little bit surprised because in vitro, this elastase is almost like bleach to a cancer cell, yet in vivo, we saw attenuation of tumor growth, but it just wasn't the efficacy we were really excited about. And so we started to think perhaps the tumor microenvironment has some of those inhibitors that might directly antagonize the enzyme. And upon organ culture and Western blots, you can see some of these inhibitors such as alpha-1 antitrypsin. Now, elastase has been studied for many, many years, but just not so much in the context of cancer. And there was an old paper showing that the porcine pancreatic elastase has a similar substrate specificity to neutrophil elastase, but better resists inhibition by these serine protease inhibitors. And you can see on the x-axis, we're titrating up A1AT, and you can see how PPE better maintains its catalytic activity and cancer cell killing capability over neutrophil elastase. And then we just did a quick pilot experiment where we injected a single dose here on day 0, and you can see with PPE, you get immediate tumor regression within a 24-hour time period. So with that data in hand, we then went into a C3 tag GEM model where we were introducing PPE or its catalytically dead version, PMSF PPE. This is a GEM model that spontaneously forms tumors in all of the mammary fat pads, but you have one that's dominant, and when that one reached roughly 100 millimeters in size, we injected it on day 0, day 1, and then never again. And you can see how nice the control of the primary tumor is with just 2 injections of the enzyme. What got us even more excited was to look at the distal tumors. And so these are all the other tumors that are spontaneously developing, both in terms of number and in terms of weight. You can see dramatic decreases at those distal tumors that never saw the active compound. And just like you saw in the previous talk, if we depleted CD8 positive T cells, we could abrogate that distal effect. I'm not showing you the data, but the primary effect is retained because that's due to direct cytotoxicity to cancer cells. All right, so to summarize, I think we've now kind of convinced ourselves that neutrophil elastase is able to overcome cancer cell heterogeneity. We've tested now over 50 cancer types and have yet to find a cancer cell that can't be killed by it. We've identified this cancer cell-specific killing program, and we're really excited about the idea that in this one enzyme, you can really combine multiple therapeutic modalities as a single agent. And what I mean by that is there's numerous therapeutics whose goal is to diminish survival pathways or to induce DNA damage or kill cancer cells. Of course, there are the immune activating approaches, but neutrophil elastase kind of combines all of this as one single agent. And finally, PPE really highlights the need for protecting it from serine protease inhibitors in the tumor microenvironment. So with that, these are the folks that help with this work, and I'll take any questions. Thank you.
Thank you very much. Outstanding talk. Do we have any questions from the audience? Maybe time for one question? See at the middle microphone, please.
Thank you for a great talk. Can I just ask about the effects on hepatocytes? Because CD95 ligand is toxic to hepatocytes, and that's one of the cell types I didn't see represented in any of your data.
I can't recall if we had tested primary hepatocytes. Yeah, I don't think we've tested them.
So I just have a crack, because it's really worth trying out, because I did my PhD in apoptosis, and that was kind of a stop-go thing for a lot of these approaches in humans. So.
Yeah, what I can share is that there's no evidence of liver toxicity when we inject these into animals, but we haven't done the in vitro studies.
Good. Right, thank you.
Yeah.
Thank you very much.